Principles of gene regulation. Two important concepts in the regulation of transcription initiation are constitutive genes versus inducible genes. In constitutive genes, they're typically on all the time. They're typically the housekeeping genes that are expressed continuously by the cell. So you can imagine things like ribosomal RNAs that are needed all the time to make ribosomes, for example. And inducible genes, on the other hand, are genes that code for inducible enzymes needed only under certain environments. So the example that I give is beta-galactosidase, which is essential for utilizing lactose. The microbe wouldn't need the enzyme beta-galactosidase if there's no lactose around. So that particular set of genes that includes beta-galactosidase would need to be regulated very specifically. Repressible genes are, again, typically encode enzymes that function in biosynthetic pathways. Generally, these are enzymes that are always present unless the end product of the biosynthetic pathway is available. So inducible genes are often part of catabolic pathways. So again, repressible genes are typically part of biosynthetic pathways, whereas inducible genes are typically part of catabolic pathways. So in this particular decision tree, you can see a couple different metabolic decisions that microbes have to make, and you can apply that to the concept of gene expression. So over here, biosynthetic enzymes, if the end product of the pathway is present, indicated here, then there's no need to synthesize more of it, and so the operon would be shut off. However, if the end product of the pathway is not present, then we would synthesize these enzymes, and so those genes and operons would want to be on. Over on this side, catabolic enzymes, it's a little bit, there's an extra little decision that has to be made here. So it's not just a matter of is the substrate of the pathway present or absent, but there's also an additional dichotomy over here. So in this particular case, if the substrate of the pathway is not present, then obviously you don't want to synthesize those enzymes. There's nothing to catabolize, so no need to waste your energy on that. Over here, however, if the substrate of the pathway is present, the decision making isn't quite done yet. The next question that the microbe has to ask, and obviously I'm being teleological and microbes aren't consciously thinking of this, but the next thing the next gene regulation type question that has to be asked is the preferred energy source or carbon source present? And of course, if the answer is no, then the operon gets turned on. If the answer is yes, there's something better to eat, then the microbe might shut things off before switching to metabolism of that other nutrient. So the classic example of that is if you take an E. coli cell and you plop it into a medium that contains both glucose and lactose, the microbe will shut down its lactose utilization genes in favor of glucose utilization. Once it's eaten up all the glucose, then the lactose utilization genes get upregulated. A good example of this is some of these concepts is the tryptophan biosynthesis operon, and it's regulated by the TRP repressor. So again, this is a series of enzymes that are required for tryptophan biosynthesis. So in this picture, you've got panel A and panel B. In panel A, there's low tryptophan levels. Transcription of the entire TRP operon occurs. And so in this particular case, the TRP repressor that you see here is transcribed and translated into an inactive repressor. And so RNA polymerase can touch down on the promoter and read through and create the entire polycistronic messenger RNA. Over here, there's high tryptophan levels. And so again, the microbe doesn't need to make tryptophan. And so in this particular case, a little bit of the tryptophan can sit on this little allosteric site here in this enzyme and basically turn it into an active repressor that can then fit right here into the operator on the operon and shut it down. The arabinose operon is an interesting example of a operon that's controlled by a transcription factor that is both an activator and an inactivator, depending on whether arabinose is present. In this particular case, cartoon A here shows operon inhibited in the absence of arabinose. So here's the Ara-C protein forming this weird connecting loop here between this operator 1 and Ara-I gene over here. There's another Ara operator over here too that the Ara-C proteins will bind to, and you get this sort of tweaking of the DNA molecule so that there's repression of the arabinose operon. Over here in cartoon B here, operon is activated in the presence, and so each of these Ara-Cs bind to arabinose, and that turns them into activators essentially. The loop is broken. This weird little loop is broken, and then these Ara-C arabinose complexes can become activators to initiate transcription. And so what you're seeing here is I threw these in just so you can see the Ara-B, Ara-A, and Ara-D genes. So Ara-A is something that will convert L-arabinose to L-ribulose. And then the Ara-B over here is a ribulokinase, which will take the L-ribulose and phosphorylate it to L-ribulose phosphate. And then Ara-D is will convert L-ribulose phosphate to xylulose phosphate. And then in this state, this particular molecule can then be shunted into the pentose phosphate pathway where the microbes can use it to make energy. Another concept in gene expression in bacteria is the use of two-component regulatory systems and phosphorelays. And so in this particular case, the main components, it's not always two, and I'll show you in the cartoon in the next slide, but there's typically two proteins that govern the pathway, but it varies. So the first protein is typically called the sensor kinase, which sort of sits in the membrane, the cell membrane, and kind of hangs into the periplasm of gram-negatives and basically will, or gram-positives, but sits in the membrane and interfaces with the periplasm. And so there's a sort of a extracellular component, or a periplasmic component rather, that's looking for signals from the environment. And that when it finds those signals, it feeds that information to a response regulator protein. Typically, the response regulator then is the DNA binding protein that can activate or repress DNA expression or gene expression. And so in this cartoon here, you see in panel A a classic two-component where you have the sensor kinase sitting straddling the cytoplasmic membrane with a periplasmic component that's waiting for some kind of signal, and then the response regulator here that's a cytoplasmic. And so in this hypothetical example, imagine a signal comes in and touches down here that may trigger a phosphorylation event where a phosphate group from ATP is transferred to a key histidine, and then this kinase domain transfers the phosphate to a response regulator, specifically an aspartate residue that elicits a response, either a negative or positive gene regulation. Over here, it's just a slightly different version, but still the same concept. This is a phosphorelay system with a connector. So we have the sensor kinase, a connector, and then an additional phosphodonor, and then the actual response regulator. So it's the same concept, phosphates moving through different proteins, but there's just two more components. So another interesting concept in gene expression in microbes is riboswitches or sensory RNAs. And so the image here, and you can read this text, but the image here goes along with that. This is a riboswitch of the rib operon from Bacillus subtilis. And so over here, in this particular, you're looking at the gene expression on mode. In this particular case, the riboflavin, this is the rib-O is the riboflavin biosynthesis operator. And so the operator is not occupied, and so the rib genes, rib E, for example, and others, which would be over here, get expressed to make riboflavin. And then riboflavin can be converted to flavin mononucleotide. And then in this particular case, as flavin mononucleotide builds up, it can bind to this little RFN box here in this leader region on the messenger RNA. And that promotes the formation of a terminator loop at the end to block transcription. So the presence of this flavin mononucleotide tells the cell that there's a lot of riboflavin and that we don't need this operon anymore. And again, I put some notes over here. Riboflavin is vitamin B2. So the cells need vitamin B2 as a cofactor for different enzymes. And flavin mononucleotide is also important in metabolism. Another example of this type of riboswitching occurs at the level of translation. I should point out that this is more at the level of transcription, right? You're dictating whether transcription starts or stops. In this particular case, it's at the level of translation. So the messenger RNA has been made, the Shine-Dalgarno, our ribosome binding site, is available, the ribosome assembles on it, here's our start codon, and then a protein can be made. But in the presence of some ligand, there's a particular binding site in the messenger RNA that would trigger another region here to bind to the Shine-Dalgarno and form this loop, which blocks ribosome binding and in turn blocks translation.